Hello, everyone, and welcome to the webinar today. We're going to be going over the formulation and manufacturing of ocular therapeutics. My name is Andrew Buis. I'm the senior formulation scientist at Altasciences, and my partner here is Anthony James. Would you like to introduce yourself?
Yeah, my name is Anthony James, I'm the Executive Director of Business Development here at Altasciences, and really looking forward to discussing, formulation and manufacturing of ocular therapeutics and this, exciting area of our industry where we're seeing a lot of growth in specifically the ophthalmic space. So excited to jump into this.
All right, perfect. Let's get into this. So here's just our headshots of both of us, because you're curious what we look like. So on the agenda today we're going to go over the importance and unique challenges of ophthalmic drug delivery, the common types of ocular dosage forms, formulating for effective ocular drug delivery, and clinical trials and commercial manufacturing. Why ophthalmic medicines matter. According to the WHO, at least 1 billion people globally live with vision impairments, typically from a preventable or treatable source. The main conditions causing distant vision impairment or blindness are listed below. And some of the more common diseases are cataracts, refractive error, age-related macular degeneration, glaucoma, diabetic retinopathy, and presbyopia.
Yeah, I think that the reason that ophthalmic medicines truly matter is because the eyes are a naturally degrading and decreasing part of our bodies that we lose our vision over time in addition to outside control, including any sort of diseases or any sort of indications. It's something that's just degrading over time. And so we know versus many other body parts that this is something that, we need our vision, we need our sight to see. And so the fact that there's so many people that are affected by this means that there's always new and innovative therapies being introduced into the marketplace to help address things that are either just related to, age related or any sort of degeneration versus things like cataracts, refractive error, glaucoma, actual indications. So I think there's a wide variety of rationale and reasons to build in ophthalmic and ocular therapies into a highlighted place in the pharmaceutical market space. But I think that these statistics really help to highlight that it affects everybody. I think a lot of us have experience with family members, friends, etc., that have these types of eye related issues. So it's one of those things where it's close to home for a lot of us. And I think it makes sense as to why. I mean, 826 million people suffer from presbyopia. So, I mean, that's not a small number by any stretch of the imagination. And I think the really exciting thing is about the recent developments in these ophthalmic medication and the recent innovation. So, Andrew, I think you wanted to cover that piece next.
So with recent developments in ophthalmic medications, as Anthony mentioned, the market is currently exploding for new drug approvals to treat some of the conditions as listed before, such as dry eye, AMD, atrophy, retinopathy, and mydriasis. So with the exploding market, many of the existing drugs are being looked at again, and and a lot of those drugs are being reformulated into new, either patent extending or new products altogether, and also including formulations with additional ophthalmic drugs to kind of provide that synergy and extend the patent life and also bring new combinations of those existing drugs to the market.
Yeah, and I think, as long as these eye indications have been out there on the market, which has been, seemingly forever, there's been drugs to treat them, right? We've had, things even as basic as artificial tears, single-use doses all the way to really intricate and complex multi-dose applications, as you're mentioning with things where you're talking about combination doses, dosing maybe two different molecules at once to get a desired effect. And I think that reformulating is something that's really interesting because although we're seeing a lot of innovator products coming out on the market where we're looking at a new molecule or an NC or something that's never been seen in our marketplace, there is a lot of space out there for reformulation and taking things that have been on the marketplace for a long period of time, reformulating those, adding new excipients, adding new ingredients and mechanisms into that formulation in order to, like you mentioned, help with patent protection. People that are going for 505(b)(2)s who are trying to use previous information, sponsors are trying to use previous information in their regulatory setup in order to advance their medicines faster through the clinical pipeline, while also allowing for, using some of that old data. Additionally, I think it's better not to invent the wheel in some cases, right? So there's something that you can do where you're tweaking the formulation, you're adding something slightly different in order to get maybe a more pronounced effect. Those things can include talking about nanosuspension of the particles, talking about, really just particle engineering at that stage, which we'll talk about a little bit later. So a lot of different innovations, both just from a base level of that molecule and in approaching something that's been around for a long time in a new and innovative way.
And many of those innovations, just to kind of tack onto that, many of those innovations actually help improve the efficacy of these drugs. Because some of the older drugs and drug compounds,
The one thing that is complex about these medications and these, the reason that these developments, there's so many over the last period of time is because of the anatomy of the eye itself, right? The way that the biology of the eye works is really different versus any sort of other type of system. You're not talking about the GI tract, you're not talking about the duodenum. I mean, those are complex in their own right, but I think there's so many different areas of the eye, as you can see on this slide and this image, that you have to take into consideration. You're looking at different anatomical barriers that are restricting the amount of medication that's actually reaching the tissues that you're trying to target, right? A lot of times you're talking about the difference between systemic and localized application of these products. The bioavailability is something that you really have to battle, as well as biodegradation, right? Things that are being biodegradable. You want to make sure that the eye is able to process those, that you're not, including excipients in your formulation that are going to be stuck at any part of the eye, whether that's inside of the outer layer, the conjunctiva, or even, talking about how we're going to pass through the blood aqueous and blood retinal barriers. These biological structures a lot of times will resist water-based drugs, and they're going to limit that absorption via tear turnover, nasolacrimal drainage, reflex blinking, and they're going to regulate, what really enters the inner eye, which causes the way that we're dosing these products to be a little bit more challenging to really make sure that they're being treated well, that they're efficacious. And in order to combat those, we've looked at, and just in the market and at Altasciences have looked at some delivery systems which we can develop to help overcome these barriers and overcome that poor bioavailability that you might see in an ocular and ophthalmic dose. So I think Andrew will discuss some of these common ocular formulations. There's quite a few. So
yeah. So I mean, the classic one is the eye drop is the standard liquid formulation system, either a solution, suspension, or an emulsion, typically applied through microliter drops, usually in an applicator, either single dose or multi-dose. You have some of the more rare ones, which also far more invasive ones. You have the injectables where the drug is injected directly into the eye or the surrounding tissue. Very invasive, very painful, obviously. And then you have your solid dose formulations, which are typically more along the lines of your gels and creams, which you're going to place along the upper eyelid or around the eye itself. And those are typically used to deliver water-sensitive drugs, providing a zero-water release profile or sustained residence time. Then you have the more systemic approach, which is your classic. You need to get your drug into the bloodstream to have that bioavailability there, and then eventually it'll make its way to the eye. However, that is very limited to the drug's bioavailability and the high dosing.
and I think although eye drops make up around 90% of the ocular formulations market, I think there's a space for the rest of these as well, just based upon the type of profile that you're looking for, the type of admission you're looking for, if you're trying to get an administration to a localized or systematic route. Additionally, ocular films, the solid dosage forms that aren't maybe brought up as much. You have your ocular films or lenses that can be either dissolving or not. You can have inserts that are soluble or insoluble, hydrogels, where you're actually adding the drug as a solution or a dispersion. And even in some rare cases for products that are affecting the eyes but can be dosed systemically, you can dose these products orally. You can also even have, in very, very rare cases, mini tablets applied to the conjunctival sac, collagen shields, and more. So a lot of different approaches, a lot of different ways to dose these products. And I think a lot of different ways to read how these products are being processed by the eye and by your overall ocular location, including aqueous humor, vitreous humor, taking those fluids, and you're really able to get a good readout on where these different molecules are being processed, how you're getting a readout on their efficacy, their bioavailability, and how they're being absorbed by the body by using those different types of approaches. There are pros and cons, though, so it's not all blue skies and rainbows. But I think just based upon the ocular challenges of how the eye's biology works, you'll see on here there's different reasons. And I think, a lot of these reasons point to why those eye drops, emulsion, solution suspensions really do make up a large bolus of the products that we see in the marketplace today. And Andrew, you want to discuss any of these specifics?
Oh yeah, I mean, like mentioned earlier, solutions are typically the easiest one, but yeah, it all comes down to what Anthony said, that you need to meet the conditions on the ground in this case. So whatever, like trying to treat, you kind of need to match that with the dosage form. So if you need like glaucoma, but you also need like a long release profile, now you can go with more of like a liposome, more of an actual structured formulation, or you can go with more a hydrogel, which increases the retention time on the eye. Now there are, like you said, there are pros and cons to each of these. It could be either processing time, it could be the complexity of the formulation, the stability of it, the contact time. It could also boil down to the cost and the general manufacturing complexity, the amount of equipment involved in that, and also the stability of the system you're trying to introduce, whether that be temperature, pH, onic strengths, or with the patient, it could just be poor patient compliance with different forms.
I think we've talked about, a lot of different formulation approaches and a lot of different dosage forms that we can look at, but it's really about how can we make sure that they're effective, right? I think on the next slide, we talk about, formulating for these ocular drug delivery mechanisms to be as effective as possible. So using different approaches for ocular drug development. Two of these main ones will be reducing the particle size and improving the bioavailability of the product. So reducing the particle size helps to deal with those physical barriers of the eye by actually decreasing the size of the particulate of the molecule to be able to improve the API's bioavailability and its ability to go through those different barriers. You're looking at a few different barriers in the process of getting through to be administered in the eye itself. And so getting to a smaller particle size, getting a lot more pieces of those particles to be able to touch the solvent system that you're dosing them with, whether that's going to be in an emulsion or a solution or a hydrogel or a suspension, that really is going to help give a huge uptick in getting the drug actually through the eye's biological barriers and getting it absorbed by the body. In addition to the particle size itself, just overall improving the bioavailability of the medication can help to increase the time during which the medication is in contact with the eye. It's a way that you can make sure that the product is as efficacious as possible. And one of those ways to do that is through nanotechnology. So a lot of different ways to do this, but Andrew, you can talk about firsthand how we do this over here at Altasciences as well.
At Altasciences, we specialize in delivery platforms through nanotechnology, typically wet milling in this case. So this is the case where you take a suspension, a particular suspension, and you grind down the particle size to a nano range. By doing that, you dramatically improve its bioavailability. And in the case of ocular medicines, it increases the retention time and the release profile of your drug substance. But this is typically useful for very hydrophobic compounds. So it helps get around the solubility issues on that. So in order to do that, the API is first milled to reduce particle size, followed by dispersing the particles in an aqueous surfactant. And the surfactants, based on the toxicology reports of an eye, have to be fine-tuned to not increase any kind of toxicity. The particle size is directly indicated drug substance in time and activity on the precorneal surface. Suspensions also require a dissolution or release of a drug substance from its suspension matrix. Now, that suspension matrix, that also increases the retention time of your drug on the eye, as well as providing stability for the formulation itself. And we get into that a little later. And also, nanosuspensions will help dramatically improve the bioavailability of the hydrophobic drug substance by increasing the solubility and the residence time. Wet milling media is just one of the technologies we have here at Altasciences. We also explore nanoemulsions and polymeric nanoparticle technology. For the emulsions, just your classic oil and water dispersion of two immiscible liquids. Typically you solubilize the hydrophobic APIs in your oil component and then suspend that into an aqueous phase, or vice versa if you have an aqueous soluble drug. These droplets are then reduced again to a nanoscale and then administered that way. And then the oil core or the aqueous core, depending if you have an oil and water or water and oil emulsion, acts as the drug reservoir, which gradually releases the drug, thus reducing the dosing frequency.
I was just going to say I think overall through all these different techniques that we're discussing, a lot of it has to rely on the surface contact of the API or the molecule with the eye. And as you and I both know, there is a bit of a challenge with having the prolonged exposure and prolonged surface contact of these emulsions, these oils, these polymeric nanoparticles, as well as even nanosuspensions and other types of products with the eye, while also ensuring that, individuals are able to see, don't have blurred vision. So not only are you trying to get the efficacy by increasing the amount of time they have the surface contact, but you also need to take into consideration that, eye blindness or things that can be, felt immediately after dosing are not prolonged, and that you have patient compliance in the sense that, people that are taking these products are not experiencing any sort of discomfort, while you're balancing, I guess, the efficacy versus the discomfort of the potential patient or the person being dosed. So I think that's something that we definitely want to take into consideration as well, just as we're discussing all these different ways that we're, helping with these emulsions and these oil-based systems to help that contact with the eyes.
So the polymeric nanoparticles can be processed very similar as the nanoemulsions, but in this case, you use a biodegradable polymer that encapsulates the drug within a polymer matrix. It can be either within the surface, or it can either be within the polymer matrix itself or on the surface, and allowing you to enable the drug release and target delivery. So with the previous technology was the solid dispersion wet milling down into nanoparticle crystals. With this technology, we're dealing with more encapsulation. These technologies, so nanoemulsions and polymeric nanoparticles, so PMPs, the APIs are encapsulated either in an oil droplet in the case of nanoemulsions or in biodegradable polymers. And the key difference with this is that these help solubilize and protect the APIs from the ocular environment. We are able to effectively control and extend the release profiles on this. In the case of the polymeric nanoparticles, we can extend the release profile for weeks, thus reducing the amount of the dosing frequency for your drugs. The lipid and the oil components also help facilitate drug transport across the corneal epithelium and the stromal layers of the eye. These technologies allow us to use the vehicle as more of an API depot, which extends the release profile and helps protect the drugs away from the ocular environments. It helps dramatically reduce dosing frequency and allows us to encapsulate both hydrophobic, hydrophilic, and for a growing market of macromolecule drugs, in the case of gene therapies, peptides, proteins, and vaccines. So prolonging the retention time with many drugs definitely increases the bioavailability of the drugs on an ocular surface. The best is a very critical strategy to further enhance their effectiveness and improve patient compliance by reducing the frequency of administration. In order to help prolong the retention time from a formulation side of things, there are several things we can do to the formulation, such as adding viscosity enhancers, mucoadhesive polymers, either formulating the products into gels and ointments, or designing the formulation in a way that it's a liquid administration, but then upon application, it gels up into a more viscous gel. So to improve the retention time, you typically so in the case of improving the retention time for viscosity modifiers, instead of having your standard eye drop.
So in order to improve the retention times, you can add different types of modifiers, viscosity improvements. So the biggest thing for that is the eye presenting this unique challenge of the physical barriers as well as the dwell time, and making sure that the product has as much contact with the eye as possible, thereby improving that through oils, any sort of surfactants, any sort of lipid dosages, to make sure that you get that molecule having as much contact with the actual target area as you can. Additionally, mucoadhesives, you can enhance the time the drug stays on the cornea by adhering to the mucus layer of the tear film, actually allowing the product to act over time. Gels and ointments, you can significantly enhance the residence time versus just purely liquid formulations. So once again, just talking about how that viscosity plays a huge role in prolonging the retention time of the molecule on the eye itself. Additionally, you can have different polymers that help transition the liquid to a gel upon application. So once you apply the liquid as a, non-viscous, non-gel type of approach, you can actually have polymers that change with temperature, pH, or ions to actually become more gel-like after you apply it to the eye. The other things you have to take into consideration when you're looking at prolonged retention in the eye is avoiding any sort of irritation, inflammation, or foreign body responses to the product in the eye. Products also have to keep their potency over a large extended period inside the eye's aqueous style environment.
So on to the fun part, the manufacturing of ocular drugs for clinical trials and commercialization. The manufacturing a topical eye medication for the commercial market involves critical considerations that are tested during clinical trials, and these factors include, and probably one of the biggest ones, is the sterility and contamination piece, as most ophthalmics need to be done in either a grade A or grade B clean room, an ISO 5 or an ISO 7 room, in order to maintain sterility as an aseptic manufacturing. But not too many facilities can have those types of clean rooms. So in the case of Altasciences, we like to manufacture in our grade C suites, and then we like to either add preservatives to eliminate the bioburden during manufacturing, or we sterile filter and terminally sterilize the product in order to keep the microbial contamination and bioburden down. Another factor is the long-term stability of the formulation. This is typically built into the formulation itself, but so by the time it gets to commercial manufacturing, the stability profile has been tested and is acceptable for a long shelf life.
I think in addition to these, understanding the analytical instrumentation that's needed varies with these types of ocular and ophthalmic dosage forms. Typically when you're seeing a solid oral dose or even an injectable dose or something similar, you're looking at significantly different analytical testing than you would for an ophthalmic product. Specifically, you want to make sure that you're looking at viscosity testing, osmolality testing, being able to understand that residence time in the eye based upon the physical characteristics of the drug product itself, as well as sometimes looking at particle size of the product. We mentioned particle sizing earlier, nanoparticles, to be able to test the particle size of those actual of the compound when it's inside of the solution. And then, like you mentioned, sterility and contamination. So being able to test endotoxin, bioburden, and even extractables, leachables on the final container enclosure can be, something that you'd want to look into when you're developing and manufacturing these types of dosage forms versus something like a tablet or a capsule where you're testing things more along the nature of dissolution. Overall, though, you still want to make sure that you're testing for assay and related substances because you want to make sure that over time your product keeps the same potency and also isn't showing any degradation or signs of, unwanted related substances in the product as it sits on stability or sits on someone's counter. So I think there's a few other ones that you wanted to touch on as well.
Yeah, they're still important, but they're not as important. So, um, continue on with the manufacturing, the things we're looking for. One of the options that's been coming up with the market is the extra scrutiny on preservatives right now. So, with the growing market for ophthalmic medications, preservative-free options are becoming more and more popular. It's effectively you eliminate the preservative system inside the ophthalmic drug, and then you terminally sterilize or you aseptically manufacture a sterile product, and then you can use preservative-free special enclosures to administer the medication. That builds into the next one, which is the container and delivery system. As you can have your standard eye dropper, which is a tube and a dropper, but then there's also special containers that help prevent UV sensitive drugs from degrading from extra exposure to heat and UV light. The upgraded containers are typically patent protected because they usually have a unique design that prevents, or I guess in this case, prevents backflow, maintaining sterility for a longer time. Um, and many of them are designed with the idea of preservative free in mind. So many of these materials are actually terminal sterilization or can withstand the terminal sterilization process of either heat, gamma, or EB.
Yeah, and I think one of the biggest things here is that terminal sterilization element. We talk about scalability and cost efficiency, running a full, aseptic process might not be needed in some cases based upon FDA requirements for phase one, especially for those first in human trials or for any sort of GLP tox or animal studies. Um, and the cost savings that you can experience versus using a terminal sterilization product versus something that's an aseptic fill finish product all the way through can be significant differences in terms of overall budget and cost. So that's a bonus there. Additionally, you can make the bulk solution in a environment, terminally sterilize that bulk solution, and then transfer that over to a different provider to do the final fill finish in an aseptic manner. So there's a couple of different ways that you can get creative with your manufacturing scalability, your cost efficiency, your budgeting for the project, in order to make sure that you're still making those high quality, you know, um, regulatory backed ingredients while also ensuring that you're not breaking the bank while doing it, which I think is a pretty key point here when talking about the different ways that you can navigate a project between preservative-free, terminally sterilized, or aseptic fill finish.
With that, our presentation is done. Does anyone have any questions?
Hey, Andrew, I have a question here. Someone wanted to harken back to the discussion about cost differences between the aseptic manufacturing and terminal sterilization routes. Can you describe a little bit more into detail about, what those differences are and why it might be a benefit for someone to choose one over the other or, differentiate between the two?
Yes, Anthony. So the key difference between, or there's several key differences between the aseptic manufacturing and terminal sterilization using an ISO 7 room. In the case of aseptic manufacturing, you require a specially validated room. The controls on the grade A suite, the ISO 5 room, are very, very strict, very compliant with regulatory. They need to be within very strict regulatory requirements in order to maintain its sterility. And the upkeep on these facilities are very expensive, talking hundreds of thousands of dollars a year just to maintain these rooms. And that's also not to explain that you still have to do the aseptic testing of it. If there's any kind of contamination, it's a much more higher risk to the product in this case, because you still need people who are highly trained to go into these aseptic manufacturing suites. And if for whatever reason the batch is contaminated, it does throw the whole thing out, and you end up wasting a lot of money. The other thing with the aseptic manufacturing is that not too many places hold these kind of facilities, so the cost just based on that alone is going to be exponential when compared to, say, manufacturing in a grade C suite and then sterile filtering and then terminally sterilizing. I can say for a fact that I have experienced both, and I will say both for the client side and the manufacturer, terminal sterilization is definitely a much safer way of doing it. It's definitely a much more effective way of doing it because you don't have to maintain the excessive costs of a grade A suite, and the regulatory restrictions are much more lenient when it comes to manufacturing a grade C suite and terminally sterilizing. So the whole terminal sterilization piece, these are all typically tested prior to getting in the clean room, so you know exactly how your product reacts to either it be an autoclave, so heat sterilization, or gamma sterilization, or e-beam sterilization. So you understand what you're getting into when you do this approach. But in terms of cost, the manufacturing in a grade C suite versus manufacturing in a grade A suite is a very, very wide gap. It is far cheaper to manufacture in the grade C suite and terminally sterilize, sterile filter when applicable, and then terminally sterilize and test your product that way.
Yeah, you answered it a little bit here, but I have a bit of a follow-up question that's being asked in terms of the terminal sterilization element. It sounds like there's testing that's done before the sterilization. Is there anything that you do after the sterilization event to ensure that the product is still maintained and hasn't degraded at all?
So yes, once the product is returned from the terminally sterilization facility, it undergoes more rigorous testing, typically alongside the unsterilized material. So that way you can have a good side by side if anything breaks down. So typically when you sterilize these products, the high energy usually it sterilizes through degradation of bacterial DNA. So, but in this case, a high energy can affect the API. So when you're running the test in comparison with the unsterilized material, you were looking for mostly API degradation and breakdowns of the delivery vehicle.
Andrew, I have a question here from somebody asking about the preservative-free trend that ophthalmic products are trending towards in the modern era. Just out of curiosity, are you seeing more of these preservative-free options? What do those look like in terms of packaging configurations? What's your, I guess, what's your thoughts and stance on the market trend that we're seeing today?
Yeah, so with the new applications of drugs, of existing new drug identities and existing products on the market, preservative free is many people want to go with the more preservative free option, as the typical preservatives for ophthalmics are pretty toxic. Talking about the benzalkonium chloride, and it's effective, but it's not ideal because of its toxicology profile. But with the trend moving towards preservative free, as mentioned before, we can't guarantee aseptic manufacturing. However, we do sterile filter when applicable, and then also terminally sterilize to ensure that sterility. And then we tend to package these into specially designed dropper bottles. These dropper bottles have a one-way flange that prevents backflow and that maintains sterility. So preservative-free, it's becoming more popular and it's becoming more practical to go preservative-free because of advances in the packaging components and because of advances like advanced technology like terminal sterilization.
All right. If we didn't get to your question today, no problem at all. Please feel free to reach out to either Andrew or I at any time. Our contact information is here on the screen. It's been a great pleasure having this conversation with everybody today and talking through, this exciting time for ophthalmic products and ocular therapies in the industry and how we can support those over here at Altasciences and across the market. Thank you so much and have a great rest of your day.